welcome to Chapter 14, Sensation. Just a quick FYI, the images in all of my PowerPoints are either from the OpenStax Anatomy and Physiology book, which is non-copyrighted or has an open copyright, or I've either made the images myself or tried to find them online somewhere where there is a, as this says here, Creative Commons copyright or open copyright. So we're talking about sensation. The first thing you want to know is that you have these special cells called receptors, so they are receptor cells, and they're all specialized neurons that respond to environmental stimuli. The two properties that I want you to know that all of your sensory receptors have is that they transduce stimuli and have the ability to release neurotransmitter. We're going to talk about what those two things mean and how it all works. But those are the two tidbits that I want you to be familiar with now. And then I'm just going to throw out these questions for you. We are not live, so you cannot answer them or discuss, but they still bear throwing out there. So you may remember from Anatomy and Physiology 1, postsynaptic potentials, EPSPs, IPSPs. If your professor didn't cover them, that is okay. Not everybody geeks out on the electrophysiology the way I do. But you may recall what causes them, what they are, and how they work. If you remember that, then I want you to know that all of that information is applicable to your sensory receptors because they're neurons too. So they're going to have a lot of the same phenomenon, or they're going to do a lot of the same things that neurons in AP1 did. The terminology is just going to be a little bit different because the mechanisms are a little different, but a lot of the underlying sort of science is the same. So you might remember that your regular interneurons in your brain have these postsynaptic potentials, which are examples of graded potentials. That means they're not all the same. So in this case here, you have this green synapse, or this green neuron, is dumping neurotransmitter on our central neuron here, and that is causing depolarization that we see in that graph there. That would be an excitatory postsynaptic potential. And then over here, these red neurons are dumping a different neurotransmitter onto our central neuron, and that's causing hyperpolarization, or an inhibitory postsynaptic potential. What I want you to know is that your sensory receptors do the same thing. So they respond to different kinds of stimuli. They might be chemical stimuli like flavor molecules like sugar and acids, which would work the exact same way as a neurotransmitter does, or it might be mechanical. So this is an example of a non-chemical form of how your neurons work. All right, so you have these structures in your inner ear, inner ear, that help you detect movement of your head and different kinds of equilibrium. It's not important that you fully understand all of this right now. I'm just trying to give you an example. So they're cells that have little hairs that stick up off of the surface of the cell. This is actually an extension of the plasma membrane. So this green part here is continuous with the cytoplasm in the rest of the cell. So it's one big fluid compartment all connected. When you move your head around, this gelatinous goop right here sloshes back and forth and causes these hairs to bend. When the hairs are straight up and down, if you look right here, that is an ion channel, and that ion channel is closed. When some sort of mechanical stimulus causes these hairs to bend, then the ion channel opens. Here they have entry of some sort of positively charged ion. Can anybody think of an ion that would flow into a cell and cause the inside to become more positive? Maybe sodium, so we'll put that in there, right? So it's just like the rising phase of the action potential, or an EPSP from Anatomy and Physiology 1. So in AP1, we called the neuron's response to neurotransmitter an EPSP, or a postsynaptic potential, IPSP or EPSP. In sensory neurons, we call the same phenomenon, this change in the resting membrane potential, we call that a receptor potential. But it's still, something happens to the neuron, its membrane potential changes. So that is called transduction, just taking the stimulus and having some sort of mechanism in the membrane that has the stimulus cause a change in the resting membrane potential. This receptor potential, as I said right here, is a graded potential that you have in your neurons. And you want to remember that they can be depolarizing, like this receptor potential here, or you could have a receptor potential that is hyperpolarizing, which would be sort of an inhibitory stimulus or an inhibitory receptor potential. Not all of this will be on the exam. I just like to give students enough information to really understand this stuff. So then this receptor potential may or may not lead to the formation of an action potential, and this is dependent on what kind of sensory neuron or receptor neuron we're talking about. In the case of this cell here, you can think of this as one of your traditional sensory neurons. So maybe this encapsulated nerve ending here is out in your thumb, right? That would mean this business end here is in your spinal cord. So that axon has to be as long as your arm to get information from your thumb all the way to your spinal cord. This sensory receptor is going to require an action potential to get information from here all the way to there. This cell down here is one of your photoreceptor cells. It's in your retina. This picture makes it look just a little bit shorter than your sensory neuron, right? But this is your more typical microscopically small cell. So this cell is hundreds of thousands of times shorter than this cell right here. So I can't come up with the math for it, but it's a lot shorter. So that any receptor potential in one part of the cell is going to have measurable effects on the resting membrane potential throughout the entire cell. So in the case of this photoreceptor here, light hits, that's supposed to be a lightning bolt of light, this part of the cell, and is going to cause changes in the membrane potential that are immediately felt there where the neurotransmitter would come out. So because the cell is so small, the receptor potential affects the resting membrane potential of the entire cell, and you don't need an action potential. What is going to happen in both of these cases is that whatever happens on the sensory end of the cell is going to change how much neurotransmitter comes out this end of the cell here. And we want to remember that it is going to be a change in neurotransmitter release rate. So it doesn't always mean that stimulus equals a release of neurotransmitter. And we will have examples later on in the later on in this chapter today, where we have sensory neurons that release neurotransmitter in the absence of stimulus, and then when the stimulus hits them, they hyperpolarize and stop releasing neurotransmitter. I'm going into a lot of detail because I'm a nerd and I love this stuff, and I want you all to have enough information to really understand it. Let's see. This is a slide that I should have deleted, so cross this off in your PowerPoints. Don't worry about it. So this is the summary of the last 9 minutes of explanation that I've just given you about your sensory neurons. What I want you to know is that they all transduce stimuli, which means their stimulus causes a receptor potential, and that receptor potential is going to cause a change in the amount of neurotransmitter that is being released. and it may or may not lead to the production of an action potential. So that's sensory neurons or receptor cells in a nutshell. Now we move on to, what do we move on to? Let's just move on. This slide I'm not going to spend a lot of time on. You want to know what adaptation means, and you want to understand what specificity of sensory receptor means. And I will point out, just because it makes the world more interesting, hot and cold, two separate sensations. Your body's thermoreceptors are not like a thermometer. They don't tell you what temperature it is. What they tell you is whether or not heat is entering or heat is leaving the body. So if heat is entering the body, it is perceived as hot. If heat is leaving the body, it is perceived as cold. An example of this in your everyday life would be you get out of the shower, you step on the bath mat does not feel cold. You step off of the bath mat onto maybe a tile floor, and the tile floor will feel cold. So I'm going to ask you a question. Assume the bath mat has been sitting on the floor all night in the same room. Is the bath mat the same temperature as the floor? The answer to that question is yes. They are both the same temperature. They've been in the same room, in contact with each other all day. The difference is that the bath mat is insulating, so it doesn't pull a lot of heat out of your body, whereas the tile floor is non-insulating. It is a very good conductor of heat, so when you step off the bath mat onto the tile floor, the floor feels cold because it's better at pulling heat out of the bottom of your feet. So there's just a little tidbit to make your showering experience more interesting, because that's what science does. It just explains the world and makes it interesting. I'll shut up and move on. This slide here, just commit these definitions to memory. You don't want to hear me talk over all of this. I will assume you can understand it and commit it to memory. I'm going to briefly talk about nociceptors because they are special and interesting, and again, they're just one of the things that makes your life more interesting, so you better understand the experience of inhabiting your own body. So all receptors, if stimulated to a great enough degree, can cause pain. So really loud sounds can be almost painful. Bright light can hurt your eyes. Nociceptors, when they get stimulated, always produce uncomfortable, irritating, painful stimuli. That is their job. And they're there mostly to let you know that whatever you're doing is damaging your body. There are different forms of nociceptors, so you can see here they can respond to physical stimuli as well as thermo and chemical stimuli. And it's the chemical stimulus responding nociceptors I'll talk about for just a second. This is why you have extended muscle soreness, or it's why bee stings hurt so much. So think about that. Like a bee stings you. The stinger is relatively small. It's not doing a lot of mechanical damage, but that venom is activating nociceptors, and it's really good at it. So it hurts like heck when you get stung by a bee. Or God forbid, a murder hornet. Have you guys heard of these murder hornets? This is crazy. But at any rate, that is nociceptors. And the other interesting phenomenon that they explain, as I was saying, is the muscle soreness. So if you go to the gym and you haven't been to the gym in a while, and you go get on the chest press machine or actually do bench press with the free weights big Olympic bar, you are going to be sore for a couple of days afterwards. This is not because several days later you still have lactic acid floating around. This is because you've torn apart some of your muscle cells, and stuff that is supposed to stay inside the cell is now outside the cell, and your nociceptors pick up on that and tell you that you have torn up your chest and you should give it a rest for a couple of days. And then the other interesting thing about the nociceptors is that the neurological pathway that gets the stimulus from your muscle to your brain works a little bit differently, and that is why ibuprofen and Tylenol and other painkillers aren't really good at blocking muscle soreness, because the neurology of muscle soreness is different than the neurology of mechanical pain. Let's see. Then we move on to classifying the receptors by their location. So commit this definition to memory, commit that definition to memory. Then we get on to another special slide, or special type of receptor called the proprioreceptor. This, as it says here, is a special type of interoreceptor. They are stretch receptors that are located in all of your muscles and tendons and joints, and they report to your brain, and basically what they do is tell your brain which muscles are stretched, which muscles are contracted, and they also provide your brain with important information about how much tension you're putting on all of your joints, so that you know when to tap out in your jiu-jitsu class, if you take jiu-jitsu. But if you think about what this really does, is that if your brain knows that your bicep is shortened and your tricep is lengthened, then you must be flexed at the elbow. So taken together, what your proprioreceptors do is give your brain a picture of what position your body is at any given point in time. And it's hugely important to being able to operate your body, because if you don't know what your limbs are doing, then you don't know how to direct them to perform movements. And it's one of those aspects of how your body works that we don't think about very often. We just sort of take for granted that we understand where our body is, but it is a sensation or a function that you can actually lose, and people do sometimes lose their proprioception because it's a different neurological pathway than regular body sensation, like hot, cold, touch, all of that. So just another interesting thing. Oh, and I forgot, the functional class here, right? This should be mechanoreceptors. I will try not to draw on the screen too much if you find that annoying, but I just think it's fun that I can do it. So this slide here, I just want to say these two terms, primary pathways and offshoots, those are my own creation to differentiate between different neurological pathways. So the primary pathway, as this says here, is how the stimulus gets to the area of your cortex that's responsible for that sensation. So whether it's somatosensory cortex, auditory cortex, visual cortex, that the pathway that ends there I call the primary pathway. Now other parts of your brain are all wired together so that, let's see, the example I have here is your limbic system, right? If you eat a bitter red plant that makes you sick, you are not going to forget that. The same way, if you've had too much of any one thing to drink, weeks or months later you catch a whiff of that same adult beverage and you're going to get queasy. So your limbic system is made aware of that, the taste of whatever it is that made you sick, but it's not part of your perception of it. That's an example of an offshoot. So then, now we are on to our first official sense, which is smell or olfaction. So you simply want to know smell is olfaction, and the organ of olfaction is your olfactory epithelium, right there on the roof of your nasal cavity. Additionally, you want to know, let me turn the page on my own notes here. First thing you want to know is that if you are going to smell something, it first has to be dissolved in the mucus of your olfactory epithelium. It will then come into contact with one of your olfactory receptor cells, these yellow things right here, that are just traditional looking bipolar neurons. So these are going to work the same way a neuron in Anatomy and Physiology 1 did. only instead of a neuron dumping acetylcholine or dopamine or a neurotransmitter on them, they're responding to chemicals that are dissolved in your mucus, like scent molecules from your soap or cologne or whatever it is you're smelling, activates these neurons the same way neurotransmitters activate neurons in your brain. So collectively then, all of these axons here are what make up your olfactory nerve, which is cranial nerve number, does anybody remember? It's number 1. I always have to review that list because I don't do well with lists. So moving right along, you just want to know the proper order in which information is going to pass through different cell types. And things get crisscrossed here. So you start at the bottom of the figure, but the top of the text. So first, your olfactory receptors are going to be stimulated or depolarized by odorants. That signal is going to generate an action potential, which travels up your olfactory nerve, and that is going to dump neurotransmitter onto the mitral cells in your olfactory bulb. And then collectively, the axons of all of your mitral cells are what make up your olfactory tract, this part right here. And that's what you saw on the underside of your sheep brain, which you may have referred to as olfactory nerve number 1, but it's not a nerve because it's outside of the central nervous system, so we call it the olfactory tract. And that goes straight back to your olfactory cortex. So your olfactory system is the only sensory system in your body that's wired straight into your cerebral cortex, because it's one of the oldest senses that any organism has. It's the oldest sense to evolve on the planet. So there was a sense of olfaction before there were these other brain areas that could relay or interpret sensations. Moving right along. So then we get to the offshoots. So you simply want to know that in addition to your olfactory tract reporting to your olfactory cortex, neurons branch off of it and end at the limbic system and the hypothalamus. We won't worry about what the limbic system and the hypothalamus do. You just want to know that those two parts of your brain get olfactory sensation information. Next up, taste. This one is straightforward as well. You simply want to know that taste is gustation, this being anatomy and physiology. We can't just say smell and taste. We have to say gustation and olfaction. You want to know that the receptor structures, really small organs for sensing taste, are called papillae. So you are probably used to talking about the structures that you can see on the surface of your tongue as being taste buds. If you can see it with your naked eye, it is not a taste bud, it is a papillae. So these are the big structures that you can see when you stick out your tongue in the mirror. If you zoom in on one papilla and look very closely at the sides of it, you will see individual taste buds, which are microscopically small. So this is one taste bud here, and oh, that color's not going to show up, sorry. I want a new color. This is a taste bud there. You can just kind of see that the cells come to a point right there. That's your taste pore that's up there. This slide will help you when it comes to lab. You're not going to see, well, you might see a picture like this on the exam, because the exams are going to be lecture and lab all jumbled together. And hopefully that'll make things easier, because you just know all of the anatomy and physiology for the exam and don't have to worry about keeping things separate. Again, know that taste molecules have to be dissolved in saliva. I don't care that you know about the gustatory hairs. That's just a tiny little detail. This list could be interesting if we had time to talk about it, but this being a screencast, I will just say commit this list to memory, because you don't want to listen to me talk over these slides all day. Whoops, let me just make sure I didn't forget anything. Yeah, I didn't forget anything. Just commit the list to memory, and yeah, we'll leave it at that. Sorry. So then the gustatory pathway, this would be the primary pathway, is cranial nerves 7 and 9. Does anybody remember which one 7 and 9 are? I have to like count with my fingers to do this. 7 is facial, if you don't remember. And 9 is glossopharyngeal, which I am not going to try and write with my little pen here. So cranial nerves 7 and 9 are going to go to the thalamus, and they're going to dump neurotransmitter on cells in the thalamus. And then cells from the thalamus are going to project to your gustatory cortex. That is the part of your brain that is responsible for perception of taste. And then the offshoots include also the limbic system and the hypothalamus, again for regulating hunger and memory and energy balance and all sorts of stuff. Nothing from this slide is on the exam, so I won't spend too much time talking about it. It is interesting to note, like right here, most of what you think you taste, most of what is perceived as taste, is actually smell, right? You've just got these 5 flavors that your tongue can detect. So it would be like if you were trying to put together a band that only had 5 members. What you taste is more like a symphony. So all of that extra flavor and nuance and great sensation that you get from eating something that tastes really good, that experience is as rich as it is because you have all of this smell sensation that you're perceiving as part of the taste. And in addition to that, right, the temperature and the texture of the food also has a lot to do with how you perceive its taste. This is why I don't want to say processed, but I'll say processed food, anything that you would get either at a restaurant or especially packaged food. This is why it tastes so good, because these companies have figured out what your brain likes and know how to manufacture foods with the right texture and the right combination of salty and savory and the right fat content to push all of the buttons that they know are going to make for a pleasurable sensory sensation. All right, moving right along then, we are now on to your sense of vision or sight. I'm not going to ask you any questions about eyebrows. The only thing you want to know about eyelids is that we give them a special name called palpebrae. That's it. Conjunctiva, you do want to know this is a transparent membrane that's on the anterior or front surface of your eye, as it says here, which produces some lubricating mucus that helps, again, lubricate your eye so that your eyelid runs over it smoothly. You don't need to know about the conjunctival sac, but I just want to point it out so that people understand one thing. So your conjunctiva runs across the surface of your face and then doubles back down here. That little part right there, that's your conjunctival sac, and there's another one up there. What this means is that if you put a contact lens in, it can't swim around to the back of your eye. It'll get caught in that conjunctival sac, and it might get wedged pretty far in there so that you think it's fallen out, and then 5 hours later it just plops on the desk while you're studying. But you can't lose contacts behind your eye because of the conjunctival sac. There you go. Lacrimal apparatus. Again, this is not super important. So know that your lacrimal gland is the source of all of your tears. And the ingredients in tears is not in your objectives, so we'll just agree to not put it on the exam. The pathway of tears as well, not super important, not an open-ended question. I don't think Oh, we did have one open-ended question. I forgot to mention this because I'm trying to be efficient. These 5 tastes here, you might have to type those 5 words into the computer on your first exam. So be aware of that. And I will try and remember to be better about referring to your objectives as we go through. But just so you can see, where is it? There it is, your 5 tastes right there. So I have the objectives out when I'm doing these screencasts. You should have the objectives out when you're studying. Moving right along, sorry about that. Yeah, so just commit this to memory. It doesn't really bear needing a lot of expectation. Basically, your tears start here, and they go across and there and down there. This, by the way, right, this nasolacrimal duct, that goes into your nose. This is why when you cry, your nose starts running. It's not snot, it's just tears that haven't run down the front of your face. They've drained into your nose. And that's happening all the time, is a small amount of tears is always being created and washing across the eye and draining into your nasal cavity. And if it's just a small amount, it goes down your throat and you're never aware that it's there. So now we're on to how does vision work. And what I'm going to do is talk about this list here as we look at this picture here. So I divide up light into, or I should say vision, into 4 different steps. This is my own outline, so don't look for it in any book. So your eye first regulates or collects light. That is the job of your iris. So the iris is the colored part of your eye. If you have brown eyes, you have a brown iris. Blue eyes, blue iris, kind of. And then your pupil is simply the hole in the middle of your iris. It's not really a structure, it's a hole in another structure. So your iris regulates how big your pupil is, and that affects how much light can enter your eye. Then you have your cornea, which is the clear covering on the front of your eye, and your lens. They both bend light, which helps you focus the light. Then you have to take the light and turn it into some sort of electrical signal that can affect neurotransmitter output, right? That's transduction. That is going to take place in the retina, the inner lining of your eye there. And then all of the raw data from your retina is going to be sent back to your primary visual cortex, and your primary visual cortex is going to take the data from your 2 eyes and put it together to give you one 3-dimensional image of the world. So then, light collection, we said, is the job of the iris. You want to know that the iris contains 2 different muscle layers, a sphincter pupillae, which some books will call the circular muscle, and dilator pupillae, which some books will call the radial muscle. I've gotten in the habit of saying sphincter and dilator pupillae. You can use either. So if you have, let's start here, bright light, or you're looking at something that's very close up, your pupil is going to constrict. The constriction happens because your circular muscle is going to contract. So these orange fibers here are going to get shorter. And when those fibers get shorter, your round pupil gets smaller and you get a constriction of your pupil. This, you want to know, is under the control of your parasympathetic nervous system. Dilation of your pupillae, or excuse me, of your pupil, is what happens when your radial muscle or dilator pupillae is contracted. So here now you have muscle fibers that are arranged like spokes on a wheel, and if all of these muscle fibers get shorter, that's going to pull your sphincter pupillae or pull your radial muscle wider, and your pupil is going to dilate. So that's step 1, is regulating how much light can come into your eye. And this is also an open-ended question. So I might ask you, how would your eye respond to bright light? How would it respond to dim light? And you would need to be able to say, constrict, list the muscle, and the branch of the autonomic nervous system that controls it. Relatively short, open-ended questions is what I'm going for on the exam so that you're not trying to type paragraphs. So now we have to focus the light. The fancy science word for focusing is called refraction. This happens when light passes from one substance into another substance that has a different density. This is why a straw in liquid looks like it is broken at the surface of the water or the liquid, because the light coming out of the liquid bends when it moves from liquid into glass, whereas the light that's not in the liquid is not refracted when it makes that phase transition. So the straw appears to be broken or disconnected. The same thing happens when light passes from air or the liquid in your eye into either your lens or your cornea. The angle at which the light hits the lens affects whether or not it's refracted, but to be honest, we don't really care about that. So let's just move on. So why do you have to focus this light in the first place? So what you want to understand is that every object in your field of view is scattering light in all different directions. So you might, if you pass by a nice shiny car on a sunny day, you get that starburst effect off of the chrome, or a really nice new paint job. That's happening all the time. You're just not always aware of it. So you always have this starburst effect or divergent light rays coming off of every object in your field of view. What your eye has to do is get all the divergent light rays from any point in space to meet again at the same point on your retina. That's what focusing is bending all the light rays back together. What you need to know in general is that the cornea does focus light. It can refract the light, but it is fixed thickness. It doesn't get thicker or thinner, so it can't adjust the focus. It's like a magnifying glass versus a pair of binoculars. Binoculars you can focus, a magnifying glass you can't. So your lens focuses or refracts light, but because it's flexible, it can change the extent to which it refracts the light or how much it bends the light. This is what allows your eye to adjust for something that's close versus distant. Before we get into that, I want to do a quick anatomy review. So here is your lens, and then you will notice that you have a ciliary body. That's this part here and that part there. It's a circle of muscle that's attached to your lens. I always use ciliary muscle and ciliary body interchangeably. There's a difference between the body and the muscle, whatever it is, the process in the muscle, but who cares? It's the ciliary thing, which is a muscle that controls the shape of your lens. So now, if an object is distant, the light rays that hit your pupil are not very divergent. So the way to understand this is to think like if you're firing a rifle and you're very far away from the target, you can't change the angle or the elevation of your barrel much at all before you're off target. But if you stand 2 inches away from the target, you can point the gun down, you can point the gun up, and it's still going to hit the target, right? So as objects become closer, they can be more divergent but still hit your lens, which means your eye is going to have to focus or bend light rays down here more than up here, right? So if it's distant, you have to bend the light through this smaller angle up here, and if it's close, you have to bend the light through a much greater angle. So the way this works is your ciliary muscle is attached to your lens by ligaments, and when your muscle is relaxed, right, all of the fibers that are arranged in a radial fashion get longer, which means the muscle that is a circle becomes a wider circle. And that just sort of elastically pulls on the lens. So it is just the elastic relaxation and elongation of your ciliary muscle fibers that pulls all around the circumference of your lens, which is flexible, and that causes the lens to stretch out and flatten. When your ciliary muscle contracts, right, the circle gets smaller. That eases the tension on the ligaments that are connected to the lens, and then the lens relaxes and assumes its naturally more rounded or bulged shape. So your lens, left to its own devices, wants to be kind of rounded, and when your ciliary muscle relaxes, that pulls it thin. This gets a little confusing here because this is one of the few times when a muscle is actually going to lengthen and cause something to move. Usually we're used to, with our skeletal muscle, contraction equals movement, but now it's the relaxation that is actually pulling the lens flat. So let's get back to how your eye works for distant versus close-up. This is an open-ended question on your exam, and I have decided that what I'm going to do is break it down into sort of smaller open-ended questions. So how does your ciliary muscle work for something distant versus close-up? Or how does your lens work for something distant versus close-up, and why? So you're not going to have to explain all of it, but you want to know how each structure in the eye we're going to go over changes from distant to close-up vision and why that change is necessary. So starting with something that's distant, right? Now the light rays hitting the lens are practically parallel, not very divergent, so the lens doesn't have to bend the light as much. This means that the lens can be thinner. So your ciliary muscle is going to relax. This pulls the lens thin. All of this is under the control of your sympathetic nervous system. At the same time, your pupil is going to dilate, right? Because you're looking far away. You're trying to get a view of things in the distance, and you want light from your peripheral vision so that you're seeing as much of the world as you can. And that's why your pupil dilates when you're looking at stuff far away. When you change to something that's close up, three different things happen: accommodation, constriction, and convergence. So for close-up vision, for distant vision, everything is on the same slide. For close-up vision, it's spread out over a couple of slides. So first up is accommodation, right? Now the light rays are more divergent, which means your lens needs to be thicker if it's going to focus the divergent light rays on the same spot in your retina. So now your ciliary muscle contracts. This allows the lens to bulge, and you focus the more divergent light rays. At the same time, your pupil is going to constrict, because now you're just looking at what's right in front of you. You don't need light from the periphery as much, because the periphery is not as important to you. And the more light that you have entering the eye, that sometimes bounces around or scatters a little bit in your eye, and if you let more light in, it actually cuts down on your visual acuity. So to get a sharper view of what's right in front of you, your pupil will constrict. At the same time your pupil is constricting, your eyes are going to converge. This picture here is obviously an exaggeration of that. I have no idea who that kid is. I just found him on Flickr, but he's not copyrighted. So there, he, she, that little child is there with the crossed eyes. So, right, divergent light rays require accommodation, constriction, and convergence and understand all of that. This slide here, we're just going to skip because I never asked questions about it, so who cares? And this slide here, just know what astigmatism and cataracts are. I am not going to explain them. They're just sort of simple definitions. This here we can talk about a little bit. I don't want to spend a ton of time on it. And yeah, this is going to be our last topic for this screencast too, because it's getting long. Where am I? Emmetropic. This is normal vision. So if you have normal 20/20 vision, what the 220s mean is that you can read from 20 feet what everybody else can read from 20 feet. If you are myopic, this means you are nearsighted, so you can see things that are in front of you, but you cannot see things far away. What's happening here is that you're actually over-focusing the light, and it's either because your lens is too thick or your eye is too long, but either way, the light rays are converging before they hit your retina. So it's divergent light rays again that are hitting your retina now. So that is myopic. You can see close up and not away. So if you have bad vision, or not as good as average vision, right, 20/40, that means you have to be a maximum of 20 feet away what other people can read from 40 feet. So 20/40, the 20 always means you can read from 20, and the second number means where can other people stand and still read it. So if you have, say, 20/15 vision, that means you can read from 20 feet what the average person has to move up to 15 feet to see. So that's all visual acuity, which we address briefly in lab with the Snellen eye chart. That's the chart that's on the wall when you go to get your eyes checked, with all of the letters with different lines under them, the red line and the green line. Hyperopia, this is if you are farsighted. So you can see things that are far but not close up. This is because you're not properly accommodating, so you're under focusing the light, and the light rays are still divergent at the point when they hit your retina, and the focal point is actually behind your retina. This I should just delete. It's the same picture. Then we are going to stop there because 47 minutes is too long for me to keep talking. Stay tuned for part 2.